Welcome to our lab podcast on the lymphoid system. And as we started out in lecture, we're going to start with our diffuse lymphatic tissue. And in this case, we're looking at that mucosal associated lymphatic tissue. And depending on where it's located, you could also call it gut associated, which is GALT with a G, or bronchular associated, which is BALT with a B. But understand, we're still talking about mucosal associated lymphatic tissue. In this case, we're in the respiratory tract, and we're looking at the lumen of our respiratory tract. Here is our cartilage of our airway. And here we're going to be looking in this area. We have some glandular tissue, but some of this increased density is going to be accumulations of lymphocytes. So again, here's our hyaline cartilage. We have some muscle here. This is going to be our epithelium, which when we get to these organ histology, we're going to refer to this as the mucosal layer. And here you can begin to see accumulations of cells, and that's going to be our diffuse lymphatic tissue or our mucosa associated, the MALT. But in this case, since we're looking at respiratory tract, we're going to be talking about the bronchiolar associated lymphatic tissue, or BALT. Again, higher magnification, we can see our nice isogenous groups of our cartilage. We can begin to make out our pseudostratified appearance. Here we see some glands and ducts of glands, and then we have our accumulation of lymphocytic tissue. Again, these are transient structures. They're not always going to be present, and certainly not in the same location, but we can definitely tell that these are our lymphocytes. Again, higher magnification, all these very small, dark nuclei, very characteristic of lymphocytic infiltrates, or in this case, our diffuse lymphatic tissue. Now, our encapsulated lymphatic tissue begins with our lymph nodes. And in our lymph nodes, we're going to see that in the hilum, we have a lot of very thin-walled vessels, no red blood cells present here. These are going to be our efferent lymphatic vessels. We're going to have our capsule on the outside, and we actually can see some of our subcapsular sinuses, that subcapsular space there. And within some of these, we're going to see other lymphatic vessels, and this would be the location of our afferent lymphatic vessels. The darker region where we're going to see our lymph nodules is the cortex, and the lighter region that's going to be our medulla, where we have our medullary sinuses and our medullary cords. So again, looking higher magnification, we can see the cortex, the outer capsule of our lymph node. We've got some trabeculae diving down. It's kind of hard to see those in this particular image, but very easy to pick up our secondary lymph nodules with a lighter germinal center. And here we have our medullary sinuses and medullary cords of cells between. Here's a higher magnification looking at our medullary sinuses. A lot of lymphocytes here, the smallest of the cells. We have our larger granulocytes, macrophages, antigen-presenting cells also swimming through here. But notice we don't have red blood cells. So that's going to be a tip-off. Even at this high magnification, we're looking at a lymphoid sinus. And here one of our cords. And since we're talking about the lymph node, this is going to be medullary cord and medullary sinus. Now, in this particular image, I believe we're taking a high mag look at a germinal center that we see here on the right. And this is going to be the outer, darker corona or mantle portion of our secondary lymph nodule. So we see a lot of lymphocytes. Some of the larger cells are going to be the antigen-presenting cells, macrophages, reticular dendritic cells. And in some of these areas of the lymph nodule, as we're getting out of the cortex, moving more into the pericortical region, we're going to see certainly blood vessels. We can recognize our endothelial layer. But in some of these, the endothelium are going to be high. This is going to be our high endothelial venule. And we actually have two, I believe these are lymphocytes that are present in the lumen. And this one, you see how it's almost teardrop shaped. This one is either squeezing out of or squeezing into this high endothelial venule. That's fairly characteristic of a shape of a cell that's undergoing diapedesis. Just can't, in this one, I can't tell which direction this particular cell is moving. Now, in this particular image, there probably is enough information here for you to determine what this organ is. And as we do, we're going to see our connective tissue capsule. We have certainly our lymphocytes here. We've got our subcapsular space. We can see a trabeculae diving down. And then out in this capsule, we can see a vessel. And we see the leaflets of this vessel. No red blood cells, practically only endothelial cell thick, a lot of connective tissue surrounding it. So this is going to be an afferent lymphatic vessel present out in the capsule of a lymph node. Now here we have another image showing our capsule on the outside. We've got our subcapsular space here. Now the capsule, you can see it very clearly diving down. And as it dives down, we are going to continue this space or sinus running along with the trabeculae. Some call this a trabecular sinus. I've always heard it as a paratrabecular sinus because it goes all the way around. And this is going to divide that cortex into different compartments. Here we see a huge collection of lymphocytes, possibly a germinal center of a secondary lymph nodule. It's hard to tell at this particular magnification, but I'm assuming as we back up, we're going to see this as a lighter germinal center. All right, shifting gears a bit, and hopefully when you look at this image, you recognize a difference immediately. We're no longer in lymph node, but the organ that we're looking at here, if you'll recall back to our lecture podcast, this is thymus. We can see the outer darker area with the lighter inner medullary area, the outer darker cortex versus the lighter medulla. We can see the lobes and segmentation, so this is thymus. Now, as we look even higher magnification, again, the outer darker cortex, the lighter medullary area. But in this location, we can already begin to see these little concentric whorl-like structures, even though we don't see them in a lot of detail. Do you recall what we call these? These are Hassel's corpuscles. And as soon as you identify a Hassel's corpuscle, you know you're looking at thymus. Once again, just looking at the cells that we have present here, these are going to be some of the reticular cells, not necessarily a Hassel's corpuscle, but we're going to be looking at some of these areas in higher magnification as we look at the most dense in the cortical region. We look at some lighter areas as we move into the medullary region and sort of a paracortical type of region. So here we are, higher magnification, looking at the thymocytes, the T cells that are present here, and you can see the higher density toward the cortex. The lower density with our reticular fibers here have a large cell, likely, when we see these largest cells, likely going to be macrophage or antigen presenting cells. And then here we have the whorl type epithelial reticular cells. I believe we'd go ahead and call this a Hassel's corpuscle. So even at this high mag, we should have a clue that we're looking at the thymus. Once again, just the density that we see different present out in the cortex. And here we're looking in the paracortex into the medullary region. One interesting thing in this particular field, we've got our vessels running along. You've got some connective tissue around here, basement membrane, but we also have these cells that are lining this. These are the epithelial reticular cells that are sheathing these vessels in the cortex. Remember, these are immunologically isolated cells in this compartment from the bloodstream. But once we get out into this paracortex and medullary region, we lose that sheathing, and now we just have the unsheathed blood vessels out in this region. Again, just showing a collection of cells that we have here. The darker, smaller cells are going to be our thymocytes, T cells. We may have some B cells, plasma cells present here, but mostly we're talking about the T lymphocytes. And most of these are undergoing their processes of development because we have a number of these that are in different stages of mitosis. You can see these mitotic figures that are present here. We've got some larger cells, which are going to be dendritic cells, reticular cells, possibly some macrophages present here. We've got a sort of a bubbly appearance. And again, the macrophages, remember, are going to be those antigen-presenting cells present throughout the thymus. Again, a couple of cells in mitotic figures. We've got some macrophages present here. But the other thing that I want to point out, you see these really dark nuclei. I mean, we're almost talking about orthochromatophilic here. These really dark, small, black nuclei, these are called pycnotic nuclei. And pycnotic nuclei are indicative of dead and dying cells. So remember, in the thymus, this is where we're going to screen these cells. And if they don't pass the muster, if they don't bind with the correct affinity, or if they recognize self-antigen, they're going to be signaled to undergo apoptosis. And apparently these cells have done so, and they are in the process of dying to be removed by cells such as macrophages. All right, shifting gears again, moving into the spleen. And when you see a low mag shot, which looks like the skin of a leopard or a cheetah, your first gut instinct should be, I am looking at the spleen, looking at the darker white matter, white pulp, and looking at the intervening tissue, which is the red pulp. And at this level, you can really see a lot of those secondary lymph nodules present in the spleen. You see a lot of trabecular blood vessels, the larger ones where you see a lot of the white space. That's really the bulk of what we're going to see at this magnification. We have our connective tissue capsule, so this is an encapsulated structure, and then we have a lot of our red pulp just underneath. Once again, as we look higher and higher magnification, I believe this is one that we have seen in our lecture podcast. We see our white pulp, and then we see our central arteriole associated with many of these secondary lymph nodules. And understand that the area immediately around that central artery, that's going to be our PALS, our periarterial lymphatic sheath. Not the entirety of that secondary lymph nodule could be considered PALS, although I think in some textbooks they go back and forth. The whole thing is white pulp, but the PALS is that particular part that ensheathes and goes around those vessels. Yeah, here you can even see periarterial lymphatic sheath, and they're just highlighting just that is the periarterial lymphatic sheath. Here, that's just the PALS. The rest of this is secondary lymph nodule. Here we have a secondary lymph nodule. I don't like that they call this a splenic nodule. I mean, I would rather you say secondary lymph nodule in the spleen, because that's really what we're talking about. And there's our higher magnification looking at that central arteriole in this case. We've got our secondary lymph nodule here, our PALS, the area just immediately around. This is a fairly small one, so we could theoretically include this whole thing, but I still like the cells immediately around that arteriole to be considered our PALS. See, they do the whole thing. I think that's a bit much based on what I've seen in other literature and textbooks. Once again, we see our radial arteries. We saw this one in our lecture podcast. This is going to be coursing through our white pulp. This is like the germinal center. Here we have sort of our coronal region of the white pulp. Where marginal zone and the red pulp starts and stops, that's very difficult to tell. That's just a very gray transitional area, but we can certainly see our red pulp in this particular image. Here's another view showing our white pulp, and here this is a little bit different because this will be our central arteriole, but then here you see these smaller vessels sort of clustered together. These are some of our penicillars, And this location is a little bit tricky because you can see it within this secondary lymphoid nodule. And so we've got the pals here. We've still got some pals here, but this is in our white matter before we get out into the marginal and our red pulp here. Here we have an interesting, somewhat tangential section. We've got some connective tissue. That's really what we're looking at. So this is a trabeculae. You can see an endothelium on the inside. So this is going to be a fairly good-sized muscular trabecular artery. Here you can see those smooth muscle cells here in the media. We've got our trabecular connective tissue out here, sort of continuous with the adventitia. So that's going to be a trabecular artery. Now, this is a neat high magnification slide showing those splenic sinuses or venous sinuses. We can see some of our endothelial cells lining these, our staph cells. These are the nuclei. But here we can see again that teardrop-shaped red blood cell, and you can see that this is squeezing out of the sinus. There you can see one that's squeezing out. Here's two trying to get out. There's one sort of in midstream, and we can see that throughout all of these splenic sinuses. These red blood cells are going out into the space that's the cords of Billroth. We're going to have other sinuses that are going to collect these in sort of an open circulation way, drain them into the venous system to get them out of the spleen and back into the circulation. Now, I really don't like this term. You see migrating red blood cells. Red blood cells don't die of Patis. They're just being forced with hydrostatic pressure through those sites. So I don't really like that term. Now, these next couple of slides, you've eliminated the soft tissue, the cellular component, and we're just looking at the reticular fibers, the collagenous network that is left. And in this image, you can see the white pulp. You can see the central arterioles around the white pulp. And out in the red pulp, you can see where we're going to have those splenic or venous sinuses. It's really kind of a neat picture. Here we can see these perpendicular fibers. This would be our splenic sinus running in this direction. So the cellular components would be parallel in this long plane, but there are reticular fibers holding those staph cells together. So this is a splenic sinus. Here we have a splenic sinus. There we have another splenic sinus. And the space between would be where we have the cords of Billroth, or the splenic cords. Now, finishing out our encapsulated lymphoid tissue, we once again are looking at palatine tonsil. Again, this is what we saw in lecture. And we're going to expect to see the oral mucosa on the free surface. We're going to expect to see 10 to 12 or so tonsillar crypts. Now, in the plane of section, again, like this one, we just see one. But I hope you can get an appreciation for how much larger this is than what we're going to see in a lingual tonsil. And that really, that size difference in scaling is going to be a big way that you tell the difference between the one smaller crypt and a smaller organ on the side of the tongue versus this humongous structure that's back in your the back of your pharynx. So again, looking, we can see our secondary lymphoid nodules. We can see how the epithelium is diving down. And again, the debris that is present in this larger crypt. When the crypts are very small, like in the lingual, you're not going to see a lot of debris. But these are fairly open diameter crypts, and so you'll see a lot of cellular debris down in the middle like you see here. And again, the epithelium we're going to see on the free surface is stratified squamous, non-keratinized, and I know this looks a little bit keratinized here, but we do see some nuclei out here. And we're also potentially going to see these lymphocytes infiltrating into the epithelium itself in some of these areas of our tonsils. Now, here's something that we haven't talked about yet. What we're looking at is a section through the alimentary canal. And as you can see, we've got different layers that are present here. And again, we'll talk about those as we get to our GI tract. But out in this layer, we can see these very prominent secondary lymph nodules. And these are permanent structures. These will always be here. And when you see these secondary lymphoid nodules and the other characteristics that we know as alimentary canal and really small intestine, then we're going to know that these are called Peyer's patches, secondary lymphoid nodules called Peyer's patches, and they are present in the segment of the small intestines. It's called the ileum. And again, these are permanent structures. They're not temporary, much like we saw with our diffuse lymphatic tissue. Once again, higher magnification is very clear. These are secondary lymphoid nodules. We can see the villi of the intestine. Don't worry about those right now. We will be describing all these different layers of gastrointestinal tract, but you can clearly recognize those secondary lymphoid nodules. Those are going to come in very handy as we're looking at organ histology and gastrointestinal tract. Now, here we have one of the nodules of Peyer's patches, and you can see the lymphocytic infiltrate of the lymphocytes extending up into the epithelium and up into these intestinal villi as they're projecting up there. We can see all of these hollowed out spaces here. Our goblet cells, you're probably familiar with that, our columnar epithelium. So you should be pretty comfortable already with much of what we're going to talk about with gastrointestinal tract when we get there. This is in some of our connective tissue as we're underlying the mucosa, and here we can see some small vessels, just endothelium. Once again, here we're getting into more of our arterioles and venules. Even though we don't see cells, we might see some. No, I don't think that's red blood cells in there. But these are going to be our lymphatic vessels with all of these lymphocytes, very thin walls built in down in this connective tissue. Here's another section through one of our lymphocytic capillaries. Now here's another section through alimentary canal, and again we've got secondary lymph nodules, and in this particular instance we've got a much smaller luminal diameter, and often in this particular section we're going to have a lot of debris present in this lumen like we see in our tonsillar crypts, and this is going to be what it looks like in the appendix, the vermiform appendix. Again, we have secondary lymph nodules that are going to be permanent residents in this area with the appendix. And again, that's going to be another way that we can identify this histologically, our organ histology, in our alimentary canal. Higher magnification, again, these secondary lymphoid nodules are going to look exactly what we've seen in our other lymphoid tissues. It's just we're going to see these crypts, these intestinal type crypts, and in most instances, we're going to see debris present out in the lumen, and that's going to let us know that we're looking at the appendix.